Paternal nutrition. How a father's diet shapes his child's health. Growing evidence reveals that a father's health before conception can have a lasting impact on his children's metabolic well-being. While maternal nutrition has long been the focus, research, particularly in animal models, demonstrates that paternal supplementation with omega-3 fatty acids, fish oil, may improve offspring health. This is reshaping our understanding of heredity, epigenetics, and the intergenerational effects of diet. Why paternal health matters. For years, it was assumed that a father's diet only contributed genetically. However, epigenetics shows that nutrition alters sperm biology, influencing early development and long-term health. These dietary changes in sperm affect gene expression. Nutrients modify which genes are activated or suppressed. Embryonic development. Early growth signals are shaped by sperm's molecular cargo. Metabolic function. Early changes can influence how offspring regulate glucose, store fat, and respond to inflammation. Omega-3s, EPA and DHA, are crucial due to their anti-inflammatory effects, lipid metabolism support, and vital cellular communication. Key findings. Mouse studies show metabolic improvements. Animal studies reveal that male mice consuming fish oil before conception produce offspring with significant metabolic improvements. Improved glucose regulation. Offspring exhibit better glucose tolerance, lower fasting levels, and enhanced insulin sensitivity. Healthier body composition. Mice show reduced fat accumulation, a better lean-to-fat ratio, and lower inflammation in metabolic tissues. Enhanced lipid metabolism. Offspring demonstrate greater lipid oxidation, efficient triglyceride metabolism, and reduced dyslipidemia. These benefits stem from epigenetic reprogramming of sperm, altering DNA methylation, mRNA expression, and chromatin structure. These changes influence metabolic gene activation during embryonic development, supporting evidence. Beyond the primary research, independent rodent studies confirm similar benefits. Reduced inflammation in offspring, healthier liver function, improved mitochondrial performance, lower obesity risk on high-fat diets. These findings strengthen the idea that paternal omega-3 intake builds cross-generational metabolic resilience. How fish oil works. Mechanisms of intergenerational benefits. EPA and DHA exert benefits through anti-inflammatory effects. Reducing systemic inflammation in fathers improves sperm quality and epigenetic stability. Sperm membrane composition. Omega-3s integrate into sperm membranes, enhancing motility, integrity, and the molecular cargo delivered to the embryo. Epigenetic modulation. Fish oil alters chemical markers regulating gene activity. These epigenetic signatures influence offspring metabolic programming. Human implications. Promising, but not proven. While mouse studies are compelling, human biology is more complex. Researchers caution that results are not directly applicable to humans yet. Human studies are needed to confirm the magnitude of impact, as lifestyle, environment, and genetics also play roles. What the current research shows, emerging rodent studies continue to support the idea that paternal omega-3 intake can influence the metabolic health of offspring. For example, several recent experiments involving obese male mice supplemented with fish oil before conception have reported healthier outcomes in their offspring, including lower body weight, improved insulin sensitivity, and a reduced tendency toward fatty liver development. These findings offer valuable insight into how paternal diet may shape sperm epigenetics and intergenerational health. However, researchers emphasize that animal models do not perfectly mirror human biology, and results cannot be directly applied to humans. Evidence in humans, still very limited at present. No published human randomized controlled trials, RCTs, or observational studies have specifically examined whether paternal fish oil supplementation affects offspring obesity risk or epigenetic changes. Most available human research on omega-3s focuses on maternal intake during pregnancy or breastfeeding, with mixed outcomes related to child growth, cognition, or metabolic health, while broader studies show that a father's overall diet quality and obesity status may influence the metabolic risks of future children. These associations do not isolate the effects of omega-3 fatty acids. Conclusion: paternal fish oil supplementation shows promise for shaping future generations' metabolic health, at least in animal models. More research is needed to determine if these benefits translate to humans. For now, omega-3 supplementation remains beneficial for male cardiovascular health, fertility, and inflammation control. The emerging science highlights the importance of paternal nutrition in reproductive and family health, though further human studies are critical to confirm long-term effects.